Because I teach a course in genetics, I have a lot of videos on my YouTube channel about different forms of inheritance, which go through them in great detail. However, for some students, going through it in that much detail isn't of interest, but rather maybe a quick overview of inheritance might be preferred. So that's what this video is meant to do, to just kind of give a quick overview of different forms of inheritance. And if you should want more information, like I said, I have many videos which go through these in greater detail. We typically start with Mendel. Mendel, in his garden in the mid-1800s, figured out what no one else had in thousands of years. For thousands of years, people have been asking, why does this individual have this trait? What determines this trait? Mendel figured it out that no one in thousands of years could. So kudos to Mendel. If we were to apply Mendel's thoughts in his pea plant experiments to humans, this is what would have happened. That if you had two individuals who, say, varied in this one aspect of their outward appearance, their phenotype, and let's say that her family, whatever the blue trait is, all of her family is blue, and his family, whatever the yellow trait is, all of his family is yellow. That if they then had kids, that there would be the observation that all of the kids, might turn out, well, either blue or yellow, but in this example, let's say blue. So the yellow trait seems to disappear. Now, in Mendel's day, people kind of had the idea of blending. You would just kind of blend all of the traits together. But Mendel observed, wait, that's not what I'm seeing. And he came up with a principle of dominance. When there are these two forms of a trait, one of them is dominant over the other. One can hide the other, that the offspring then have, one trait, but not the other. And then if one were to then look at, obviously in Mendel, he could take pea plants, which were those offspring. Obviously in humans, it's a little different. In genetics, we breed fruit flies. But if two individuals who came from such parentage, say one parent had the blue trait, from a blue family, one parent had the yellow trait from a yellow family, both of them share that in common. Then when they have children, what one would notice is that the majority of the next generation, the grandchildren in this case, would have the blue trait, some would have the yellow. So the yellow kind of disappeared for a while and came back. Now, most people would stop at that. But Mendel, and this is where Mendel was brilliant, he said, wait a minute, I'm counting all of them, and it always seems to be a 3-to-1 ratio. What could explain that? How could I arrange this so that the grandchildren gives us this 3-to-1 ratio? And Mendel said that would work if, and this was Mendel's model, that for each of these traits, there are 2 units of inheritance, which we call genes. So there are 2 genes in the man that determine this trait, the color, and 2 genes in the woman. And there are different forms of the genes. So we now call them alleles. So the woman would have 2 alleles of the A gene, which we're going to represent as a capital, we'll see in a second. And then the man has 2 different alleles for the A gene. Mendel said, if they have offspring, there must be a way that even though they have 2 alleles in their cheek cells, their bone cells, their brain cells, their body cells, that when they make sex cells, their sex cells must just have 1. So Mendel was predicting that this man would give either this little a or this little a to this daughter, but not both. That this woman would give that big A or that big A to this daughter, but not both. That even though the body cells have 2 alleles, when they make sex cells, they must just have 1. Mendel was predicting how chromosomes move before anyone knew what chromosomes were. Kudos to Mendel. And so thus all of the next generation would have 1 of each of the 2 alleles. So they're different. We call that hetero. So they would be heterozygotes, having 1 allele from their mother, 1 allele from their father. These are heterozygotes, while each of the parents would be homozygotes. Both of their alleles were the same. Now, you don't know this until you watch the next generation, but if all the next generation are blue, then apparently the blue allele is dominant over the yellow. You don't know that until you observe. So now this woman would be homozygous dominant, homozygous because she only has one type of allele, homozygous dominant because she only has the dominant allele. This man is then homozygous recessive. And in the next generation, if two heterozygotes were to have offspring, then just by random chance, then some of the offspring, a quarter on average, would be homozygous dominant and have the blue phenotype. Half would be heterozygotes and have the blue phenotype, but 1/4 would be homozygous recessive and then have the yellow phenotype. So that is what is known as Mendelian inheritance. And as I just talk about it here, we could represent this in a Punnett square as well. So once again, kudos to Mendel because one, he made scientific observations of genetics after it had been mostly superstition and folklore for thousands of years. He predicted the action of chromosomes before anyone knew what chromosomes were. He made genetics a science in his garden. So kudos to Mendel. And there are many human traits, which are Mendelian traits, where there are dominant alleles and recessive alleles. So blood type A is dominant to blood type O. The alleles that would allow someone to roll their tongue are dominant against the ones which prevent you from rolling your tongue. The alleles for having a free hanging earlobe are dominant to those for an attached earlobe, etc. So we see certainly Mendelian inheritance, not only in pea plants, but he discovered traits in inheritance in general, not just peas, but also in fruit flies and also in humans. These are universal rules of inheritance. And now clearly this applies to humans. So if you ask, why did this rare genetic disease, or this rare genetic trait appear in this individual? And the answer is, well, because the mother was a heterozygote, the father was a heterozygote, and then on average, 1/4 of their offspring would receive a recessive allele from the mother and a recessive allele from the father. And that's why this rare genetic trait popped up in this family. So some human traits follow this form of inheritance. Now, like I said, there are normal traits. So, rolling your tongue, whether your earlobe's attached, whether you have hair on your mid digits, if you do, it's dominant. If you don't, it's recessive. Whether you have a V in your hairline, what's known as a widow's peak, that's dominant. Having a straight hairline, that's recessive. Blood type O A is dominant over blood type O. So there are examples of normal traits that follow this. But then certainly there are genetic disorders which follow these traits. So there are some genetic disorders which are dominant and some which are recessive. A lot of the recessive traits are when when the gene codes for a hormone or an enzyme. And so, for example, if there's this hormone which controls weight gain. It lets adipose cells tell the brain that there's enough adipose stored. It's called leptin. If one can't make this hormone at all, so if someone were to receive a mutation from both their father and the mother, where they can't make leptin at all, then they can't tell the brain that the brain has adipose, or the body has adipose. And so the brain then thinks it should be hungry when it shouldn't. And so this can lead to weight gain. And so this is an example of a genetic disorder which follows a Mendelian form of inheritance. And there are many examples, and once again, I have lots and lots of videos. There are some traits which can even affect personality. When we talk about making neurotransmitters, perceiving neurotransmitters with receptors, reabsorbing neurotransmitters, if you don't make the protein, then you know, there's one aspect that you can't, you know, perform. So there are a number of genetic disorders which are recessive, including some forms of cancer. Let's say there's a protein which prevents cancer. Well, if your mother's copy of the gene doesn't work, and if your father's copy of the gene doesn't work, then you can't make this protein that prevents cancer, and therefore that's a recessive form of cancer. Some genetic disorders are dominant. And so there are multiple ways that Alzheimer's can be caused, but a couple of the mutations are dominant, such as the presenilin 1 and 2 genes. These are dominant mutations which cause Alzheimer's. So in a dominant disorder, the heterozygotes are affected. Now, we could see that if we looked at just the proteins which accumulated. So even a heterozygote, even though half of their genes were normal, half of their genes cause these abnormal plaques to develop. And like I said, this video will go through all of the scenarios. So then even heterozygotes would be affected. So Mendel described traits which were dominant or recessive, and we know of both normal traits and abnormal traits, you know, from disorders to cancer, which are dominant and some which are recessive. Now, beyond that, there's a bunch of things that Mendel didn't observe that we have learned afterwards. Obviously, Mendel didn't discover every form of inheritance in every organism in his garden studying peas. And so there are what are known as exceptions to Mendelian inheritance. In some of these exceptions, heterozygotes would not be blue in their phenotype nor yellow in their phenotype, but actually partway in between. All right, so that there's an intermediate phenotype that a heterozygote displays, and if two heterozygotes have kids, then a quarter of their offspring have the blue phenotype, a quarter have the yellow phenotype, but then half have the yellow phenotype. There are lots of examples of that. I'd like to just quickly provide two. One involves heart disease risk and the LDL receptor. Now, if one were to study, say, the normal allele, let's call it blue here, and the, oh, my fault. Let's say that the normal allele is yellow and the mutant allele is blue. Then if two heterozygotes have kids, then individuals with two versions of the normal allele, they have normal cholesterol ranges in their blood. If you have two of the mutant allele, you have extremely high cholesterol levels in your blood. But if you're a heterozygote, it's in between. So notice normal is 75 to 175. This excessive amount is over 450, but the heterozygotes are 200 to 250, higher than these homozygotes, but lower than these homozygotes. So here's an example of incomplete dominance where heterozygotes are in between the two conditions. Now, just so you know, this is, as you study genetics in an introductory sense, it's nice to say, oh, this is incompletely dominant, this is dominant recessive. The reality is it's complicated. Some things depend on how you look at it. So if you were to ask, how do I classify this based on serum cholesterol? There are three categories: normal, excessively high, and intermediate. But if we were to just say normal heart disease risk or elevated heart disease risk, then notice the pink individual, the little a, they have the normal risk, but everyone else is elevated. Now, some more elevated than others. But here, you could say that this then is a dominant mutation, because everyone who has at least one big A has an elevated risk. So sometimes the name that you give it depends on what aspect you're looking at it. If you look at it this way, it's dominant. If you look at it this way, it's incompletely dominant. Just, if this were ever to become, your field of study, there's, more complex nuance here. A second important example of incomplete dominance involves malaria resistance and the sickle cell allele. So normal red blood cells need a specific shape to make them flexible so that they can flow through capillaries. If you change the hemoglobin protein, then you can have these cells sickle, which means they're more likely to clog capillaries, which can cause heart attacks and strokes even in children. So if each of your parents gives you a sickle allele, your red blood cell morphology changes. And that can be, a horrible genetic disease if you are homozygous for that, that mutant allele. However, if you are a heterozygote, what happens is your red blood cells look like this most of the time, but under extreme conditions, they look like this. And so therefore, you could have a healthy individual, say, join the military and all of a sudden start, jumping out of airplanes. And under those low oxygen conditions, their blood cells look like this and they have a stroke. So there is a distinction between normal individuals with two normal hemoglobin proteins, sickle cell anemia patients who have two genes from one from each parent for this trait. And then there's the sickle cell trait, where their red blood cells look like this most of the time, but under extreme conditions, they can look like this. So once again, that's an example of incomplete dominance. Sickle cell is also interesting because heterozygotes have an increased way of surviving malaria. And so that's why the allele is so common, and my videos go into that in greater detail. There are other exceptions, and once again, this is just a quick overview. So if an individual gets blood type A alleles from their parents, from, say, their father, and blood type O alleles from their mother, then we see that A is clearly dominant to O. So here, each individual has an A allele, each individual has an O allele, but they're all blood type A because A is dominant to O. In the same way, if all of these individuals get a B allele from their parents, from their father, and an O allele from their mother, then all of them are blood type B. B is dominant to O. So A is dominant to O, B is dominant to O. So far, nothing surprising. But if one parent gives a child an A allele and one parent gives a child a B allele, then the children are blood type AB. So here's another exception to Mendelian inheritance. It turns out that A is not dominant to B or vice versa, that they are co-dominant. And the reason for that, and this will go through it, is A is dominant to O because even if you only have one A gene from one parent, you will put yellow triangles on your red blood cells, all right? And so all of these children are blood type A, even though they only have one A allele coming from their father, because they put these yellow triangles on their red blood cells. In the same way, as long as you have at least one B allele, you will put green triangles on your red blood cells. However, if one of your parent tells you to put yellow triangles on your red blood cells, and one parent tells you to put green triangles on your red blood cells, and then you do both, all right? And so therefore, neither is dominant to the other. They are both expressed at the same time. The word for that is co-dominant. So co-dominance is a form of inheritance. The heterozygotes don't have one phenotype or the other. They actually have both. We actually then see another exception to Mendelian inheritance here called multiple alleles. In Mendel's original scenario, there were two alleles for each gene. So like a big T and a little t. The pea plants could be tall or short. The flowers could be purple or white. But because there's such a big population, there's all of these possible alleles. So there's A and B and O, that's more than two, and many minor alleles. If you were to study, the genes, which, say cause cancer, and ask, so how many BRCA alleles are there? There are hundreds of alleles known in the population. Some are normal, there are multiple normal alleles, some are severe mutants, some are minor mutants, etc. And so multiple alleles is another exception to Mendelian inheritance, even though each individual may only have two alleles. Nevertheless, in a big population, then you could change this amino acid to that amino acid at spot 266. You could change that amino acid to that amino acid at spot 296. Proteins have lots of spots, lots of places for amino acids, and so thus that's a lot of room for different mutations. A couple of other exceptions to Mendelian inheritance. So for example, something known as pleiotropy is when one mutation can cause multiple effects. So already with hemoglobin mutations, that would affect not only the shape of red blood cells, but would also affect your resistance to malaria. Those are two different traits, but caused by the same variant of the gene. So one gene is affecting multiple traits. That's pleiotropy. Individuals with Marfan syndrome are tall, thin, nearsighted. They have long limbs and a weak aorta. If you have cystic fibrosis, the thick mucus that your body produces can block sweat ducts, interfering with sweat, but it also blocks digestive ducts, interfering with digestion. This thick mucus can interfere with respiration. It can end up causing the loss of the vas deferens in males. And so there are different traits which can be caused by this one gene. Waardenburg syndrome, one gene mutation can cause deafness, widely spaced eyes, and a white forelock. And then this one can cause being bulbous, toothless, and the inability to sweat, which I guess I forgot to put the text for. But in any case, that's what's known as pleiotropy. And so in essence, just you have a big body. And just because these cells are using a certain gene doesn't mean that these cells can't use the certain gene. And then if you have a mutation or a different allele, that could give you like a different phenotype here, but it could give you a different phenotype here too. And so the same gene can be used by multiple sites in the body. So that gives rise to pleiotropy. Another interaction is that the environment needs to be considered. So in many of my courses, we go into heart disease. There are genes which can cause heart disease, but then there are environmental factors which can cause heart disease, like diet and stress, and et cetera. There are genes which can cause asthma, but there's environmental factors which can cause asthma, like exposure to pollution. There are genes which can cause weight gain, but clearly one's diet can affect weight gain. There are genes which can cause cancer, but there are chemicals and other things which can cause cancer. So environment can influence phenotype. It's not just genetic. And so once you know that there are genes and there's environment, now you have to consider, well, how do they interact? So for example, here are two individuals who both have xeroderma pigmentosum. So they both have a genetic mutation. This one gets cancer, this one doesn't. Why? Well, because this one avoids sunlight and only plays at night, for example. This one goes out during the day, can't protect themselves from UV light. So phenotype is not only does the environment have a role, but you have to know, the genes and the environment. So knowing both that they have xeroderma pigmentosum and whether they're, out in sunlight or not is needed. Two individuals with PKU could differ in their phenotype. All of us, when we're born, they take a drop of blood and they test us for PKU because you now have a genetic mutation. And if you eat a normal diet, then that could result in retardation. But if you go on a special diet, well, then you can be fine. And so these two individuals could have the same genotype, but because they have a different phenotype, a normal diet versus an abnormal diet, that could vary. Host of other things. So, if we were going to talk, say, about substance abuse, there are certain receptors in the brain that this makes you more prone to be addicted to substances. Now, nothing will cause you to be addicted to substances. I will never be addicted to cocaine if I don't ever try cocaine, despite the fact I have a version of the dopamine receptor D2, which predisposes me to addiction. There are liver enzymes, which may or may not break down alcohol in different ways. And so therefore, my being prone to alcoholism might be prone to what form of liver enzymes I have. But once again, if I don't drink alcohol, I won't be susceptible. And so the environment clearly plays a a role. And then very often you have to know, which combination. So with heart disease, asthma, weight gain, cancer, it's complicated. Which genes do you have? Which environmental factors do you have? Which combinations do you have? Because sometimes a good gene can compensate for a bad environmental factor, et cetera. One of the classic examples of environmental influence is temperature in Siamese cats. Siamese cats make more pigment where their body is cooler. So if you change the temperature in which a Siamese cat is raised, you can change its color, because the cat doesn't have a gene for, say, making brown feet. It only has a gene that says make brown pigment where the body is cooler. So depending on how cool the feet are. So environmental factors can play a role, genes can play a role. And one of the things that makes it complicated is many genes or many traits are polygenic. So when we consider autism, first let it be said, we still don't understand autism as well as we should. So we know many things that influence autism, but if you were to ask what's the primary cause of autism, that still is unknown as far as I know. But if you were to ask, where are there genes which affect autism risk? Well, here's images of the 23 human chromosomes. And it turns out there are lots of genes on lots of different chromosomes. So one form of inheritance is what's called being polygenic, being influenced by many genes. So here you can clearly see that. Cancer is affected by many genes, not just this one. All right, heart disease is affected by many genes. Weight gain is affected by many genes. So that's typical. And so this idea of being a polygenic trait. Now, because in the sex chromosomes, women have two of the big ones, and men have a big X and a smaller Y, this then affects a type of inheritance known as sex linkage. Because let's say there's a recessive mutation on this X. This woman will be normal. She will not express the mutant phenotype because this X has a good copy of the gene. And so therefore, while she's a carrier and can pass this along to her sons, for example, she doesn't have, let's say, color blindness or hemophilia. So she is not colorblind. She does not have hemophilia because the normal gene on this X chromosome compensates for the bad one. However, in this man, because his Y chromosome is so small and has so few genes, nothing on the Y can compensate for a mistake on the X. And so if he has one mistake on the X, then he does have color blindness. He does have hemophilia. He does have muscular dystrophy. And so some traits are called sex-linked because males are much more likely to express them. 10% of American women are 10% of American men are colorblind, but only about 1% of American women are colorblind. So gender can affect traits. So here's an example of sex-linked traits. Now, just a quick correlation going back to fetal development. This was kind of a surprise, even though you would think that the number of embryos at conception would be half male, half female, because half would be fertilized by, sperm which carry this chromosome, half by sperm which carry this chromosome. Actually, it's not half and half. And the only reason that has ever been proposed is that Y-bearing sperm are just a tiny bit lighter than X-bearing sperm because the Y chromosome is smaller. Now, what difference could that make? Well, for a cell that's swimming the length of the vagina, the uterus, and the oviduct, now uterine contractions help, but nevertheless, for a cell, that's a really long distance to swim. Having one lighter chromosome apparently makes a difference. And it turns out, and this is all over the world, all studies indicate that for every 100 female zygotes, there are about 130 males. So at conception, males are more common than females. And it seems to be because the Y chromosome bearing sperm swim a little faster. Now, when it comes time to birth, there are still more male infants than female. There's about 108 male infants for every 100 female infants. And if you ask, okay, so there were more males at conception, but wait, it used to be 130 to 100, now it's 108 to 100. What happened to 22 male embryos and fetuses? And the answer is that most miscarriages are male. When you get to puberty, the gender ratio is about 1 to 1. all right? For about 100 females, there's 100 males. And then if you ask, well, if at birth it was 108 to 100, and now at puberty it's 100 to 100, what happened to those 8 male? And the answer is that most infant deaths are male. Why? Well, there could be multiple reasons. A major one, however, is women who have a mistake on the X chromosome can compensate for it typically, where it doesn't cause the death of the embryo, it doesn't cause the death of the infant. But this would then lead to complications and health problems in males. And so the fact that this small Y chromosome can't compensate for mistakes on the X chromosome is the primary explanation why most miscarriages are male and most infant deaths are male. Now, if one were to study genetics, one could study genetics for the rest of one's life. And so this is just an overview of the things which cause inheritance. Just a couple of other points. Sometimes when chromosomes are segregating, mistakes are made, and so that the chromosomes are not split evenly the way they should. And if you look at the chromosomes that should be present in a typical human cell, there's 46 chromosomes, 2 of each of the 23 kinds. But sometimes a mistake is made where one chromosome is missing, or there's one chromosome too many, or maybe there's 2 that are missing, or 2 chromosomes that are too many. Now, this is relevant to cancer, because if you look at cancer cells, very often they have abnormal chromosome counts compared to normal cells. But this can also then be the cause of human abnormalities and genetic disorders. Sometimes this happens at conception. So for example, an individual with Down syndrome has an extra chromosome 21 in all of their body cells. A woman who has Turner syndrome is missing an X chromosome in all of her body cells. But this could also then affect parts, like in this part of the body, there's a chromosomal abnormality, but not in this other part, because the chromosomal abnormality occurred partway through development. And so if you were going to study genetics and talk about what determines a trait, very often it is chromosome number, so having too many chromosomes, too few chromosomes. Sometimes it's the change of the shape of a chromosome. So a chromosome could be missing a piece or have an extra piece, or piece of one chromosome could now break off and fuse to a different chromosome, or could turn 180 degrees upside down. So there are chromosomal deletions, duplications, inversions, translocations, which can affect traits as well. One final thing is, while most of our DNA and most of our genes are in our nucleus, in those 46 chromosomes, technically though, we still have another one, because the nucleus is the largest organelle of the cell. The second largest organelle is the mitochondria, and it has its own chromosome. Now, this chromosome is much smaller, but nevertheless, there are genes on it. And so therefore, some people have mutations in their mitochondrial chromosome, which then determines a trait. So some people have cancer because a mutation in the nuclear genes are promoting abnormal growth. But some people have cancer because it is a mutation in the mitochondrial genes, which are affecting growth. Now, since sperm tend to leave almost always, I think always, but I think there's sometimes exceptions, their mitochondria in the midpiece outside the zygote. All of our mitochondria come from our mother. And so this is a different type of inheritance where, because we're getting our mitochondria from our mother, whatever her mitochondrial genome was is the genome which we have. That is another form of inheritance. So just in general, if one were to ask, why does this person have this trait? Obviously, genetics is a broad field of science that many people spend the rest of their lives studying, because there are, it's complicated. Mendel did begin the field of genetics with, the discovery of dominant and recessive traits, but there are so many other ways that traits can be inherited and things that influence genetic traits that it's quite complex. This has been an overview of them, but like I said, in many of my videos, I go through this in greater detail. So if this wasn't sufficient detail, then I hope some of those other videos can provide the detail that you need.